check, the aims of this one are to be able to describe the formation of urea in the liver and what we call the ornithine cycle. Let's think first of all what we've covered in a previous video about the functions of the liver. Well, first of all, the liver is involved in the production of bile. Secondly, we looked at the use of the liver in the synthesis and control of cholesterol and lipids, then looking at how it removes particulates at small parts of old cells and old red blood cells, the control of blood glucose, how it can regenerate. We mentioned this previously, in fact that it can regenerate a significant amount of the liver lobes, between 6 and 12 months it takes. How it makes red blood cells in developing baby or fetus. Now in this video we're particularly interested in detoxification and also looking at breakdown of hormones. Okay, now there's one really important function of the liver we haven't yet mentioned, and that is in fact the breakdown of amino acids. Okay, so it's a very, very important function I'm going to consider here. If you are a healthy person, you need between 40 and 60 grams of protein a day to stay healthy. Obviously these guys here building muscle will require more of that, but you generally require 40 to 60 grams. Now the issue is we have no storage mechanism for excess protein. So what we're going to do about it, because it is a bit of a problem. And the problem is excess protein is toxic due to the basic amine. Amine group is the NH2 group. This is based around the functional group based around NH3. So NH3 is ammonia down here. Okay, so the amine group is NH2 and it's based around ammonia. Now, what can we do about it? Well, what we can do about it is as follows. We can actually extract the useful bits. It'd be very, very wasteful to excrete the entire molecule. The sensible thing would be to strip off the toxic bit, that's the amine group, the NH2 group, and just excrete this. And this is exactly what the body does by a process called deamination. So in deamination what you're doing is stripping off the NH2 group from amino acids. The rest of amino acid, basically it's carbon, hydrogen, some oxygen, can be readily oxidized or recycled. Now here's a basic process. It's a two-stage process. The first stage, you've got your amino acid here, and what you're going to do is strip off the ammonia, leaving behind a keto acid. Now the keto acid itself is pretty useful in terms of what it's made up of, and that can go off then and be used elsewhere. The second stage is taking this ammonia and converting it to urea, and this is all part of the ornithine cycle. So we've got two parts here. We've got deamination and we've got the ornithine cycle. Now let's just summarize here the key parts of deamination. First of all, it produces ammonia, NH3. Secondly, NH3 is very soluble and toxic. Okay, so a lot of water is taken up with NH3 and can lead to dehydration. Therefore, it's important it's converted to urea by enzyme action in the ornithine cycle, and this occurs in the cytosol and partly in mitochondria as ATP is being used. Let's just pause for a moment, think about this word here, cytosol. What actually is it? Well, quick definition here. A cytosol is liquid found inside the cells. Okay, it's a part of the cytoplasm that does not contain organelles, but which does contain other particulate matter. Okay, that's a little definition there in case you've never come across the word cytosol before. Let's go back to the deamination bit, shall we? Now in deamination, here you've got your amino acid, there is your amine group, and what we're going to do, by addition of oxygen here, we're going to remove it to form ammonia over here, and leaving behind the keto acid. So in deamination, all you're doing is you're removing the amine group NH2, replace it with oxygen, leaving behind a keto acid that we can then go and use elsewhere, and ammonia. Right, let's look on. So but a big tick there, we now know what a deamination is. Deamination is stripping off the amine group, forming ammonia. Now we need to think about the ammonia, don't we? Well, ammonia will then go into what we call the ornithine cycle. Now ornithine cycle converts ammonia to urea by addition of carbon dioxide over here. Urea is less soluble and less toxic than ammonia and is transported to and dealt with by the kidneys, where it's filtered and concentrated in the urine. So urea is made in the liver. It's then transported to the kidneys, where it's converted to urine for excretion from the body. Now let's look at how this takes place. Here we got what we call the ornithine cycle. The ornithine cycle is where we remove ammonia. I'm going to start first of all over here with ornithine. So ornithine will combine with ammonia and also carbon dioxide to form citrulline with elimination of water. Now citrulline then combines with further ammonia to form arginine, again with removal of more water. And lastly, water is added to arginine to produce urea over here, but also to recycle ornithine back into the cycle itself. So we've got ornithine combines ammonia and carbon dioxide to form citrulline with elimination of water. Citrulline combines with more ammonia, elimination of water, to form arginine. Arginine combines with water to recombine to form ornithine, but also to produce urea that is then going to be transported to the kidney for excretion as urine. Okay, that's a basic ornithine cycle. So you should pause at this point and have a go at reproducing this cycle for yourselves. We can summarize it very, very quickly by saying that over here we've got ammonia plus carbon dioxide gives us urea here plus water. Okay. This is very much an oversimplification. It misses out all those different molecules involved. It's worth just seeing the bigger picture so you know what we've got. Now we can now put a big tick. We can now describe formation of urea in the liver. Yep, I think we can put a big tick there. I think we've achieved that bit, don't you? Pretty good. Now another function of the liver we mentioned earlier is in fact in detoxification. Now the liver detoxifies waste products from metabolism as well as ingested toxins such as alcohol. Mechanisms include oxidation, reduction, methylation as addition of CH3 group, and combination with another molecule. These processes are facilitated by enzymes as catalase and ethanol dehydrogenase in the hepatocytes. Let's think now about a very important thing, detoxification of alcohol. Now, alcohol is broken down in the hepatocytes by the enzyme ethanol dehydrogenase into ethanal. Okay, moving to ethanal. Now this is further broken down or further dehydrogenated by ethanal dehydrogenase to form ethanoic acid. Okay, let's look at the equation for this. Here we've got ethanol. It's converted to ethanal, hydrogen removed, and here we've got NAD. NAD combines with the hydrogen to produce NADH or a reduced NAD. Again, ethanal is then formed to ethanoic acid. Again, hydrogen removed by NAD to produce reduced NADH. And you're left then with acetyl coenzyme A over here that can be used in respiration and is a jolly useful product. So we're getting here some useful stuff from the detoxification of alcohol. Now, last bit you need to think about is something called fatty liver. You may come across this term, fatty liver. What is it? Well, let's look at the stages. Now, NAD is required oxidized fatty acids for respiration. So as well as breaking down ethanol, okay, what is breaking down ethanol, it's also used elsewhere, oxidized fatty acids. Oxidation here is removal of hydrogen. So it's required for the oxidation of fatty acids respiration. So that means if the liver's got too much alcohol detoxified, there's not enough NAD for fatty acids. Alcohol, sorry, misspelling there, look at that, misspelling. What it's saying here is that the NAD, because we're taking in excess alcohol, okay, the NAD is being used to break down excess alcohol to form ethanal and then ethanoic acid, which means there's less NAD available for the oxidation of fatty acids. Now you can see what's going to happen. We're going to get here a buildup of fatty acids, and they're going to be converted back to lipids over here. Okay, back to lipids and stored in the hepatocytes. Now, this isn't good. This is bad news. So the fatty acids that can't be broken down because there's not enough NAD, because NAD is being used to detoxify alcohol, the fatty acids are converted back to lipids, and these are stored in the hepatocytes. To say this is not good news, and you're going to end up with enlarged liver, and this is a condition known as fatty liver. Okay, so fatty liver is alcohol related, and you get there hepatitis or cirrhosis. This is all because taking in too much alcohol distracts NAD from what it should be doing, which is breaking down fatty acids. Okay, fatty acids build up, they're stored as lipids, liver becomes enlarged, and this is what's known as fatty liver, which results in alcohol-related hepatitis or cirrhosis. Okay, not good news. Right, end this video. So in this video, as well as be able to describe formation of urea in the liver, also be able to describe the roles of liver in detoxification. So thanks for watching and listening to this video. Obviously go over it again, but if you don't understand, make sure you pause the video, take notes, make sure you fully understand this, and thank you for watching, thank you for listening. I'll be back again very soon. Bye bye for now.